Hello everyone and welcome to the new session of the biology lecture series, and today we'll be solving the NCERT Exemplar questions for the topic sexual reproduction in flowering plants. So I would like to mention that all the questions in this session are completely NCERT based, and today we'll be solving the short answer type questions. So let's start with the first question. So here you have to list 3 strategies that a bisexual chasmogamous flower can evolve to prevent the self-pollination. Okay, so bisexual flowers are those flowers which contain both the male and the female reproductive structures. Means it contains the stamens as well as the pistil. Alright, and what are chasmogamous flowers? Chasmogamous flowers are those flowers which remain open. Okay, they remain open. Okay, so they promote cross-pollination. Alright, and self-pollination you already know is also known as autogamy, where the pollens from the anther are transferred to the stigma, okay, of the same flower. This is known as self-pollination. Now let's talk about the thinking process. The majority of the flowering plants, they produce hermaphrodite flowers. What are hermaphrodite flowers? Means they are bisexual flowers. They contain both the male and the female reproductive organs. Okay, and if they are bisexual, then it is obvious that the pollen grains are likely to come in contact with the stigma of the same flower. Okay, so this promotes self-pollination. Okay, and repeated self-pollination results in inbreeding depression. Okay, so inbreeding depression is due to repeated self-pollination in these bisexual flowers. Okay, and what is inbreeding depression? It is the reduction or it is a decrease. Okay, it is a reduction in fertility. Okay, or what? The productivity of what? Of the offsprings. Okay, so this is known as inbreeding depression. Okay, now the flowering plants have evolved many devices or many methods to discourage the self-pollination. Let's talk about these. Okay, a bisexual chasmogamous flower can evolve 3 strategies to prevent the self-pollination. Alright, the first one is dichogamy. Okay, so in this mechanism, the pollen release and the stigma receptivity. Okay, so you know pollen lands on the stigma. So here what happens in dichogamy? The pollen which is released, okay, and the stigma receptivity, they are not synchronized. Okay, so the pollen release and stigma receptivity should be synchronized. Then only, okay, pollination or fertilization can take place. But here in dichogamy, there is no synchronization between them. Okay, for example, in sunflower, the pollen is released before the stigma. Okay, here you can see the pollens are released before the stigma becomes receptive. So this is known as protandry. Okay, what is protandry? When the pollens, they mature first, or the pollens are released, but the stigma, which is the part of the pistil, which is part of the female reproductive organ, it is, okay, not receptive. This is known as protandry. Okay, but for example, in Datura and Solanum, reverse can happen. The stigma becomes receptive, okay, much before the release of the pollen. So it is known as protogyny. So gyne is related to the female reproductive part. So here stigma becomes receptive, okay, but the pollens are not released. So there is no synchronization, and this leads to cross pollination. Okay, now second strategy is hercogamy. So in hercogamy, what happens? The male and the female sex organs are placed at different positions. Okay, so there is a spatial difference. Okay, there is a spatial difference, okay, between the male and the female sex organs, okay, or in different direction is known as hercogamy, okay. Now, in these plants, the pollen cannot come in contact with the stigma of the same flower. All right, it has undergone cross-pollination. For example, hibiscus and gloriosa. The hercogamy is observed in hibiscus and gloriosa. All right, and in these plants, what happens? The pollen cannot come in contact with the stigma because they are located at different positions or in different directions. So this also promotes cross-pollination. Now, third point, third strategy, self-sterility. Okay, what is self-sterility? It is a genetic mechanism which prevents the self-pollen. Okay, which prevents the self-pollen from fertilizing the ovules. Okay, and what happens? How the fertilization of ovules is prevented? By inhibiting the pollen germination or by inhibition of the pollen tube growth. Okay, so this is seen in a plant known as abutilon. Okay, so this is a plant in which self-sterility is seen. Okay, the pollens are not able to fertilize the ovules. Self-pollen. Okay, you should write self-pollen. Okay, pollen of the same plant is not able to fertilize the ovule of the same flower. Okay. Now, here is a note I have given here. Now, another method to prevent the self-pollination is the production of unisexual flowers. Okay, so if unisexual flowers are produced, so obviously there will be no self-pollination. So unisexual flower, they consist of either the male reproductive organ or the female reproductive organ. Okay, so this will also promote cross-pollination. Okay, but this is not beneficial as above mentioned strategies. Okay, so this is not that beneficial. The other methods are quite beneficial. Okay, this is the least beneficial one. Okay, now it prevents autogamy but not metonymy in monoecious plant such as castor and maize. Okay, so alright, this method of producing the unisexual flowers, it can prevent self-pollination. Okay, but it cannot prevent metonymy. Now, what is metonymy? It is the transfer of the, if we talk about this, transfer of the pollen to the stigma of what? Of another flower. Okay, to the stigma of the another flower, but this flower is present on the same plant. Okay, but this flower or both these flowers are present on the same plant. Okay, this is clear. This is also a kind of cross-pollination only or self-pollination only. Although metonymy comes under cross-pollination, but you can say it is a kind of self-pollination only because the genes or the genetics or the DNA or the genetic material is same. Okay, because both the flowers are present on the same plant. Okay, so we can say it is a kind of self-pollination, but it comes under cross-pollination. Let's talk about question number 2. Now, given below are the events that are observed in an artificial hybridization. So, we are talking about artificial hybridization. Now, you have to arrange them in the correct sequential order in which they are followed in the hybridization program. So, first is rebagging. Second is selection of the parents. Third is bagging. Fourth is dusting the pollens on the stigma. Okay, and then next one is emasculation. What is emasculation? Emasculation is removal of the stamens. Removal of stamens is known as emasculation. And last one is collection of pollen from the male plant. Okay, now let's talk about this before seeing the answer. So one of the major approaches of crop improvement program is the artificial hybridization. Means it is done by man. Okay. Now, in such crossing experiment, it is important to make sure that only the desired pollen grains are used for pollination. Okay. And the stigma is protected from the contamination from other unwanted flowers. That's why we do the bagging. We do the bagging so that the stigma is protected from contamination from other unwanted pollen grains. And we do the dusting of the desired pollen grains on the stigma. Okay, and then we rebag it. So the correct sequential order of artificial hybridization is as follows. The first one is selection of the parent. So you have to first select the parents, okay, of which you have to make a hybrid. Okay. Now, second point, emasculation means removal of the stamen or the anthers from the flower, but before the anther dehisces. Okay, you have to remove the stamens. This is known as emasculation. Okay, because you want the desired pollen. You don't want the pollens from the flower. You want to dust the desired pollens. You have selected the pollens and you want to dust them on the stigma. Okay, third is then bagging. Okay, so what is bagging? Bagging is the process to cover the emasculated flower with a bag made up of a butter paper. Okay, so there is a bag which is made up of a butter paper and then you bag the emasculated flowers with it. Alright, now let's talk about the next point. Next is collection of pollen from the other male plant. Okay, so obviously pollens will be collected from a male plant only. So you collect the desired pollens. Next is dusting of the pollen on the stigma and then you rebag it. Okay, you rebag it. Why? To prevent the contamination. To prevent the contamination from other pollens, other unwanted pollens. Okay, this is clear. Now, here is a note I've given. If the female parent produces the unisexual flower, there is no need of emasculation. Okay, if the female parent, okay, they produces unisexual flowers only, means they have pistil only, there is no need of emasculation. There is no need to remove the anthers. Okay, because unisexual flower, they consist of either the male or the female. But here, if the female parent is producing unisexual flower, the female parent will produce the female flowers only. Okay, so there is no need of emasculation here. Let's talk about question number 3. Okay, now vivipary automatically limits the number of offerings in a litter. How? You have to explain. So what is litter? Litter is the number of the offerings animals produced in a single birth. This is known as litter. Now let's have a look at the answer. Let's discuss the answer for this question. Now vivipary is defined as the seed germination while the fruit is still attached to the mother plant. Okay, this is known as vivipary in plants. Now plants which grow in the marshy places, okay, they are known as mangroves. So in these plants, when the seed fall on marshy places, they cannot germinate because of high salinity and high water conditions. Okay, this is clear. Now, so in these plants, what happens? Seed germinate when they are still attached to the mother plant. Okay, so in plants such as mangroves, vivipary is seen. Okay, where the seed germinate when they are still attached to the mother plant. And litter, we have already talked about what was litter? Litter is the number of offspring produced at or during one birth of the animals. Okay, which is usually 3 to 8 in number. Alright, this is clear. Now, vivipary automatically limits the number of the offerings in litter due to the reason that limited number of egg or the ovum are produced. Okay, so during vivipary, what happens? Limited number of egg or ovum are produced and fertilized during reproductive cycle of the females. So this is how vivipary limits the number of the offerings. Okay, so how vivipary limits the number of the offerings? Okay, by producing limited number of eggs, also known as ovum. Okay, this is how vivipary limits the number of offerings, or you can say vivipary limits the litter. Okay, now let's talk about question number 4. Now, does self-incompatibility impose any restriction on autogamy? Alright, give reasons and suggest the method of pollination in such plants. So, self-incompatibility. Self-incompatibility, we already know. Okay, it promotes cross-pollination and it prevents self-pollination. Okay, now self-incompatibility imposes restriction on autogamy. This you already know. Okay, what is the reason? The reason for this is, okay, what is the reason? The reason for this may be that the majority of the flowering plant, they produce hermaphrodite flowers. Okay, so majority of the flowering plant, they produce bisexual flowers. Okay, and what happens? Because of the bisexuality, the pollen grains come in contact with the stigma of the same flower. Okay, and there is repeated self-pollination. Okay, so you can write here that bisexual flowers are produced. Okay, or hermaphrodite flowers are produced. Because of this, self-pollination is promoted. Because of this, self-pollination happens. Okay, and then repeated self-pollination. Okay, then after repeated self-pollination, means after so many self-pollination, what happens? This results, we have already discussed, this results into inbreeding depression. This results in inbreeding depression. Okay. Now, because of this continued self-pollination, okay, this results in inbreeding depression. That's why flowering plants have developed many methods to discourage the self-pollination. Okay, because of repeated self-pollination, inbreeding depression happens. So flowering plants have devised many methods to discourage the self-pollination. This we have already seen, and to encourage the cross-pollination. One of the major way to prevent self-pollination is self-sterility. This we have already seen. Okay, other was hercogamy, dichogamy. Okay. Now, self-sterility in some bisexual flower is seen, okay, if the pollen grains fall on the stigma of the same flower, germination does not occur. This is known as self-sterility. But the same pollen grains germinate when they fall on the stigma of the another flower of the same species. Okay, so this promotes cross-pollination. Okay, so it is a genetic mechanism to prevent the self-pollination. Alright, we have already discussed about this self-sterility. Now let's talk about question number 5. Now in the given diagram, you have to write the names of the parts shown with lines. Okay, so they have given you this figure. You have to label this. Alright. Now, what is this image? Okay, this image is the lateral section of the monocot seed. Okay, now let's discuss about this. Now the embryo of the monocotyledon or the monocot, they possess only one cotyledon. This you already know. So they are mono, means one. They consist of only single cotyledon. Okay, and monocots, okay, example is grass family. So grass is a kind of monocot. So in grass family, the cotyledon is called as scutellum. Okay, so monocot consists of one cotyledon, and that cotyledon in grass family is known as scutellum. and scutellum is situated towards one side. And which side? Lateral side of the embryonal axis. Alright, now at the lower end, okay, the embryonal axis has the radicle and root cap enclosed in an undifferentiated sheath known as the coleorhiza. Okay, so at the lower end, okay, coleorhiza is present. Now, what is coleorhiza? Coleorhiza is a sheath. Okay, it is a sheath that protects the radicle or the root cap. Okay, it protects the root apical meristem. Okay, now the portion of the embryonal axis above the level of attachment of scutellum is known as epicotyle. Okay, so this is known as epicotyle, above the cotyledon. One is epicotyle, one is hypocotyle. Epi is above, hypo means below. Okay, now epicotyle has a shoot apex. Okay, so if you talk about epicotyle, epicotyle has a shoot apex and a few leaf primordia enclosed in a hollow foliar structure known as coleoptile. Okay, so coleoptile is a kind of a sheath again. Like coleorhiza, coleoptile is also a kind of sheath that covers what? Shoot apex or the shoot apical meristem. Okay, so here is the labeling here. Okay, this is the lateral section of a seed. Here you can see the scutellum, which is the cotyledon present in the monocots. Here you can see the coleoptile, which is nothing but the sheath that is present. Okay, that covers the shoot apex. Here you can see the shoot apex. Here is the radicle. Okay, here is the radicle. Then root cap covers the radicle, and finally coleorhiza, which is again a sheath that covers the root cap. Okay, or you can say it covers the root apex. Now, what is this epiblast? So, epiblast is known as the rudimentary cotyledon. So, monocot, they consist of only one cotyledon and the other cotyledon, okay, which is known as the epiblast. This is a rudimentary cotyledon. This is a rudimentary cotyledon. Alright, this is clear? So, this is LS of a monocot embryo of grass. Okay. Now, let's talk about question number 6. The question number 6 is, what is polyembryony and how can it be commercially exploited? Okay. Polyembryony, you know, poly means many, means presence of many embryos. Okay. Now, what is polyembryony? Polyembryony is the occurrence of more than one embryo in a seed. Okay, in many citrus and mango varieties, some of the nucellar cells, okay, nucellar cells are always diploid. So some of the nucellar cells surrounding the embryo sac. So you know embryo sac is present within the nucellus. So some of the nucellar cells surrounding the embryo sac start dividing. Okay, they protrude into the embryo sac and develop into embryos. Now, in such species, each ovule contains many embryos. Okay, so because of this here, nucellar cells are dividing, okay, and they are giving rise to embryo. So because of this, each ovule consists of many embryos. Usually, embryos develop from where? It develops from the egg cell. Okay, so egg cell, it fertilizes with the male gamete. Okay, and then what is formed? A zygote is formed, which is diploid, and then from the zygote, what will form? The embryo. Okay, so here embryo is forming from the nucellar cells. Alright. Now, polyembryony plays a main role in plant breeding and horticulture. Okay, the plant cells obtained from these embryos are virus free and has more vigor. Okay, they have more vigor. They are okay. Now, hybrid varieties of several food and vegetable crops are being extensively cultivated, and these hybrid cultivars possess very high productivity. Okay, so high vigor leads to high productivity. Okay, and how can you calculate the vigor? You can calculate the vigor by, there is a formula. You can multiply percentage germination with the plant height. Okay, there are many formulas for calculating the vigor. All right. So if the vigor is more, then the productivity will be more. Okay. So this is the main role of polyembryony. So polyembryony uses plant breeding and in horticulture to develop virus-free plant and to develop plant which have high productivity. Okay. Now let's talk about question number 7. Are parthenocarpy and apomixis different phenomena? Okay. Now we have to discuss their benefits. Now let's have a look at the answer. Now, parthenocarpy and apomixis are different phenomena. This already know. What is parthenocarpy? Parthenocarpy is the development of the fruit without the fertilization of the ovary. Okay, so this is known as parthenocarpy. So parthenocarpy leads to the development of seedless fruit. Development of the seedless fruits. Okay, because here what happens? Okay, the ovule doesn't develop into viable seeds. So this is known as parthenocarpy. Now, this phenomena is applied for the commercial production of seedless fruit. Okay, and some examples are banana and the grapes. So in these, seedless fruits are developed and they have high commercial importance. Okay. Now, this is used for what? This is used for, or this is used in juice industries. Okay, because they require seedless fruits. Okay, so these fruits are used to extract the juice. Okay, so there is very high demand for this kind of fruits in the juice industries. Now let's discuss about the apomixis. So apomixis, you already know, it is the formation, okay, or the development of seeds without fertilization. Okay, so there is no fertilization. Okay, now during apomixis, chromosomal segregation and recombination does not occur. Okay, because there is no meiosis. Alright, there is no meiosis. Okay, recombination and chromosomal aggregation does not occur. So characters are stable for several generations. So this is the importance of apomixis. Okay, the characters will be same over several generations. There is no recombination. So due to recombination, some characters can be lost. Okay, but if you have to maintain that character, you can use apomixis. Okay. Now, second point is it simplifies the commercial hybridized production because isolation is not necessary here to produce the F1. Okay, or to maintain the parental generation. Okay, this is the second advantage. It simplifies the commercial hybridized production. Okay, and it is very easy to maintain the parental generation. Now, adventive embryony. If you talk about adventive embryony, okay, is used in the production of uniform rootstock and virus-free varieties. All right, adventive embryony. Through adventive embryony means it is also kind of asexual reproduction, asexual method. Okay. Now let's talk about question number 8. Why does the zygote begin to divide only after the division of the primary endosperm cell? This is known as PEC. Okay. Why this happens? Why the zygote begin to divide only after the division of primary endosperm nucleus or this cell? So you already know one of the male gamete fuses with the egg cell, one male gamete fuses with the egg, Another male gamete fuses with the secondary nuclei, also known as the central cell, and then it forms the primary endosperm cell. And primary endosperm cell then forms the endosperm, which provide nourishment to the embryo. Now, let's talk about this thinking process. Okay, now zygote needs nourishment to develop. First point is this. Okay, so this will help you answer this question. So first point is that zygote needs nourishment to develop. Okay, this we already know. Now, let's have a look at the answer. The primary endosperm cell divides repeatedly and forms a triploid endosperm tissue. Okay? Triploid means 3N. It contains 3 chromosome set. Okay? Now, the cells of this tissue are filled with reserve food materials. So, endosperm, you know, is a nutritive tissue. It consists of reserve food materials. And this reserve food material are used as a nutrition for the developing embryo. So, that's why the zygotes start dividing, okay, after the primary endosperm cell divides. Because primary endosperm cell, okay, it will divide. Then what it will form? It will form the endosperm. Okay, and endosperm will provide nourishment or nutrition to what? It will provide nourishment to the embryo. Okay, that's why first there is a division in the primary endosperm cell which forms endosperm, and then endosperm provide nutrition to the embryo. Okay, this is clear. Now, the embryo divides or embryo develops at the micropylar end. This you already know because egg apparatus is present at the micropylar end. Okay, egg apparatus which consists of the egg cell and the synergids. So, 2 synergids are present. Okay, and within synergids, filiform apparatus is present. So, egg cell, okay, it fuses with one of the male gamete and it forms the zygote, which develops into embryo. So, embryo develops at the micropylar end of the embryo sac, okay, where zygote is situated. Now, most zygotes divide only after certain amount of endosperm is formed, okay, because endosperm provides nourishment to the developing embryo. Now, this is an adaptation to provide assured nutrition to the developing embryo. Alright, so this is clear. Now, let's talk about next question. The generative cell of a 2-cell pollen divides in the pollen tube, but not in a 3-cell pollen. Give reasons. You have to give reasons for this. Okay. Now, this is the thinking process. The pollen grain at maturity divides and produces 2 unequal cells. This you already know. The last cell is known as vegetative cell, which has abundant food reserve materials. Okay. And it contains a large irregular nucleus. Whereas the smaller cell is known as generative cell and it floats in the cytoplasm of the vegetative cell, which is spindle shaped and have the dense cytoplasm and the nucleus. Okay. Now, in over 60% of the angiosperm, the pollen grains are shed at the 2-cell stage, okay, which is the vegetative cell and the generative cell. In the remaining of the species, the generative cell divides mitotically to give rise to 2 male gametes, okay, before pollen grains are shed, okay. So now at 3-cell stage, what all 3 cells are present? One is the vegetative cell and other are the 2 male gametes, okay. So 1 vegetative cell and 2 male gametes, they form the 3-celled stage, okay. Now, in 3-celled stage, the pollen grains further germinate on the stigma to produce the pollen tubes. Okay. Now, the contents of the pollen grains move into the pollen tube. Okay. And what happens? The pollen tube then grows through the tissue of the stigma and style and reaches the ovary. This you already know. Now, in plants where pollen grains are shed at 2-cell stage, the generative cell divides and forms 2 male gametes later, okay, during the growth of the pollen tube in the stigma. Okay. Now, question number 10. In the figure, those have given you a figure, you have to label the following parts: male gametes, egg cell, polar nuclei, synergids, and pollen tube. So this is the figure. This is very straightforward question. They have given you the image of the embryo sac, and this is the micropylar end of the embryo sac. Okay. Now, the following are the parts of this figure. This you already know. Okay. Here is the central cell, which is formed by the fusion of the polar nuclei. This is the egg cell. These are the synergids. This is the filiform apparatus within the synergids. Okay. Here is the pollen tube, which is entering through the micropylar end. Okay. Here are the male gametes present in the pollen tube. Alright. So this is the entire egg apparatus. Egg apparatus consists of the egg cell, synergid, and the filiform apparatus. Okay. So this is clear. Alright. So this was the end of this lecture. We will continue with the rest of the questions in the next lecture.